Hepatitis C. The meaning of hepa means liver, and the meaning of itis is inflammation. It is also known as HCV, and HCV is an infection in the liver that causes inflammation and can be spread through blood and other bodily fluids. A couple ways it's spread is through shared needles and unsterile piercings and tattoos, but there's multiple other ways that it's spread. It is estimated that about 2 to 4 million people in the U.S. currently experience hepatitis C. HCV can eventually lead to cirrhosis, which is when inflammation turns into permanent scarring on your liver. Symptoms of HCV can include fatigue, fever, and jaundice, as well as body aches, nausea, loss of appetite, and stomach pain. However, many people go years without noticing or experiencing any symptoms. Once they begin to experience these symptoms, the liver is usually already very damaged. A normal pathway in the liver is called the JAK-STAT pathway. This pathway helps in cell regeneration, hepatic proliferation, and multiple metabolic processes. These jobs are all super important for keeping the liver healthy and functioning. HCV uses both autocrine and paracrine signaling. Autocrine signaling is when the cell secretes messengers that bind to the same cell receptor, so it is signaling to itself. But in paracrine communication, it is local signaling where the cell releases a molecule to send to other small distances to affect another cell, so it signals to its neighbor. This pathway starts with a cytokine known as IFN-A, type 1 interferon. A cytokine is a signaling protein that controls inflammation and helps the immune system to fight off any germs and infections. JAK stands for Janus kinase, which are tyrosine kinases, and there are 4 types: JAK1, JAK2, JAK3, and TYK2. Then, STAT represents signal transducer and activator of transcription. It's a transcription factor, and it translates signals from the cytokines. The pathway takes place on the membrane of a hepatocyte, which is a liver cell. The IFN-A release is triggered by immune cells. IFN-A is important because it warns other neighboring cells of the virus. IFN-A binds to type 1 interferon receptor, which is made up of IFN-AR1 and IFN-AR2. The receptor is activated and it can bring in tyrosine kinases from the JAK family. JAK1 and TYK2 are bound to the receptor, and JAK1 and TYK2 dimerize. Now the TYK2 and JAK1 can phosphorylate. Then the STATs enter, and both STATs carry SH2 domains, which have the ability to connect with the phosphorylated tyrosine sites of the receptor. Due to the close location of the STATs, they can be phosphorylated by the JAKs. Then STAT1 and STAT2 dimerize. Interferon regulatory factor 9 joins the STATs. Together they form the ISGF3 complex. With a nuclear localization signal, the complex enters the nucleus. Then the complex binds to specific nucleotide sequences, which are known as ISREs. This creates interferon-stimulated genes, which are antiviral genes. They protect the liver, allow for healthy regeneration, and keep a regulated immune system. However, when HCV is present, HCV proteins take up the receptors that the interferon A needs. HCV core proteins are also bound to the STAT1 and reduces the amount of phosphorylated STAT1. So this means that there's less ISGF3 complexes that are there to bind with the ISREs. This means that there is less antiviral genes that are created. HCV blocks transcription of interferon-stimulated genes and makes a less antiviral state.